Hey, Xinyu, I think we can start. Dr. Andreas has joined.
Hello.
Hey.
You
Oh,
can
no,
share
I
my
think
screen.
Xinyu will introduce you. Sophia, can you close share screen? Okay. Xinyu, are you around?
Yeah. Can you hear me?
Yes, I
Yeah.
can.
Okay.
Trying to figure out how to share my screen. Okay, here it is.
Yeah, sure. Yeah. Maybe I can begin first. So hello everyone. Hello again. I'm Xin Yuan from Milan, and I'll be your host for this keynote session. We are very excited to introduce our keynote speaker, Dr. Andreas Loukas. Dr. Loukas received his PhD in computer science from Delft University of Technology in 2014, and then doing research as postdoc at Berlin Institute of Technology, MIT. Since 2021, he joined the Prescient design as a senior principal and machine learning lead. Prescient's design is a crucial part of Roche and belongs to the machine learning for drug design department within Genentech research. Dr. Loukas is an expert in the foundations and application of machine learning to structured problems, particularly in graphs or constraint context. Many of his early works shed light on theoretical analysis of neural networks. Now he had been leading a team on generative protein design using machine learning and has brought us excellent work as like AbDiffuser, one of the few first diffusion models for joint generation for full atom protein structure and sequence. Today, I think Dr. Loukas will share his expertise and insights with us. Let's give our warm welcome to him.
Thank you very much for the kind introduction. Indeed, it's been a bit less than 2 years since I joined Prescient Design. And over the last 2 years, we've been thinking, along with many talented people, about how to create molecules, create antibodies that target disease using generative AI and geometric deep learning. So today, what I want to do is to report from the trenches about things that I found out. I decided to, rather than focus more on the machine learning contributions and ideas, I thought I should spend a large portion of the talk explain to you why this is a good problem to be thinking about and what are the fundamental challenges there. Right. So, okay. Right. So on the left, you'll see a sketch, a cartoon of an antibody, and this is I like to say that this is our the molecule of our obsession. It's a really important molecule and protein because it protects us from the world. So without it, all of us would be dead. So as you may know, right? So our body is attacked by all sorts of pathogens all the time, and as the most as part of our immune response, we use antibodies in order to fight disease. And we found that actually we can harness antibodies in order to also do our bidding. And indeed, a large fraction of the drugs that are approved every year for different diseases are based on antibodies. So we mainly use antibodies for diseases where two things happen. The body cannot itself act, and so we want to kind of step in and change things, but also the action should happen outside the cell. Okay, because antibodies do not penetrate the cell. There are three main types of diseases where antibodies are used. Autoimmune diseases are a big one, like Crohn's disease, kidney transplants, asthma. We have cancer is a very important one. There are all different types of drugs right now for cancer based on antibodies. And a prominent one, for instance, is for breast cancer, and I'm referring to it because it will come back later in the presentation. but many other types of cancers are treated with antibodies. And I don't know if you've been following the news, but we were very happy last year because finally, after a long streak of failures, we had some success in the fight against Alzheimer's, where the result of the clinical trial showed some improvement in the cognitive decline of patients. The drug in question was an antibody called lecanemab, which targeted the amyloid beta plaques in the brain of people. And so even further, there is reason to believe that antibodies will become even more powerful drugs. There's something that's called ADC, antibody drug conjugate, that combines antibodies with small molecules. And it's very exciting because it acts like a biological missile. So it has the specificity of antibodies, but also has the high potency of small molecules. And I have here, I would like to quote some news from the beginning of November that I think it merits saying. It's rare to get a standing ovation at a scientific conference, but on October 22nd, the Thomas Poles received 2 standing ovations because he announced results showing that since the 80s, a disease that hadn't been improved since the 80s, specifically bladder cancer, advanced bladder cancer, showed significant improvement using ADCs, with the mortality rate falling down more than 50%. It's really a great time to be working on antibodies. Okay, so taking a closer look, this is a slightly more refined cartoon of an antibody. You'll see that it has a distinctive Y shape, and the bottom is called the constant region, the FC, and the 2 arms, they're called the FABs. They can be identical or not, but for the purposes of the talk, we can assume that they're copies of each other. And in fact, we care more about the arms, and we care more about the tips of the arms, which are called variable regions, because these are the ones that vary, right? These are the ones we design usually. In fact, they're composed of 2 chains, the heavy and the light chain, and they also have the part that touches here the antigen, the protein of interest. These parts are loops, and they're called CDRs. And the base with these beta seeds that they're called a framework region. Okay, so in fact, what we care most about are the tips of the arms of the Y shape antibody. Again, these are cartoons. If you would look at an antibody in a bit more detail, you would see quite a bit of complexity. There are a lot of atoms there, and many degrees of freedom that allow us to play with, but also a lot of constraints that one should account for about how this bond should move. Okay, so I don't want to go too much into depth about on this, but basically inside us we have a generative model that allows us to create new antibodies that target disease. And what pharma does is, when people want to design new antibodies to, for instance, to create a drug, they try to use this biological generative models to harness them for their purpose. This usually goes like this. You have a animal, a rabbit, a mouse, a rat, can be other things, that's immunized with a protein of interest. And then we wait a week or so, and then we look at their blood and try to find within that blood proteins, the antibodies that were emerged due to the pathogen in question. And then there's a kind of a procedure there where people try to identify which are the best of these antibodies, because we have billions of them in our blood, and which of them have the best properties, and both with respect to their function, their binding affinity and expression, but also with respect to the properties that will make them good drugs. And there are quite a number of them that one should care about. So people have been thinking about how to solve this problem in different ways for a while now. And before machine learning, people were focusing a lot on physics-based approaches. I mean, they still do, but now they want to combine the two. So people working on this problem with a computational mindset encountered three main difficulties in their work. The first is that the space of antibodies is a vast combinatorial discrete space. And that space, unfortunately, is very sparsely functional. So most of the sequences that make antibodies would not bind to the target that you want, that you care about. So you're looking for a needles in a haystack, effectively. The third difficulty is that the function that we care about really depends on dynamics, so entropy, and solvent. So how basically the molecules interact with the water around them. And this is something that's very important especially for antibodies, where the interaction is happening through loops, and perhaps less important in other types of protein-protein interactions, where there's more of a lock and key kind of mechanism for interaction, right? Unfortunately, we don't know very well how to handle entropy in water and how to simulate it precisely, and that hinders us from understanding their function very well. So to show you some evidence that physics-based approaches struggle, I have tried to gather a few results from the recent literature. So on the left, I have the Spearman correlation between, this is a semi-empirical force field, Rosetta or Fold X, these are two variants. And we're measuring the correlation between this semi-empirical force field energy with binding affinity. Now, the result here is around 30 or 37%, which it seems not to be so bad, but actually in this particular experiment, we're looking at SCEMP. So these are cases where we know exactly the crystal structure, and we have introduced, and we know that it binds, and we introduce a very, very small number of mutations, 1 or 2 usually mutations, and then we're checking whether we can predict whether this small mutation, this small perturbation will affect binding. And even in this simple case, we cannot do better than 37%. The second plot shows that docking, it's still unreliable. Despite the amazing progress, usually you need at the order of 1,000 tries to get an acceptable solution out, which is 1,000 times more than we usually want. And on the right-hand side, you might have heard about free energy perturbation. These are methods that have shown correlation with experimental measurements for small molecules. Unfortunately, these MD methods, molecular dynamics methods, are too slow for any reasonable application to proteins and antibodies. So we cannot rely on them too much. Okay, so that brings us to machine learning, and of course, what we care about is, how can we do better with machine learning? There are different variants of the problem of antibody design and antibody discovery, but today I want to focus on one that I think it's more geometric in nature and fascinates me the most. And that's the following. We are presented with the target structure. And this is what the biologists have told us to look at. And somebody has painted a part of this structure with green in this case, and we're told to create a binder, an antibody that can be a good drug eventually that targets that region, right? So if you have a good solution for this, then great, you have reduced the need for animal experimentation, but you've also really increased the throughput of the way that of the pipelines of drug design within pharma. Because there are a lot of, even if you have the ability to harness biology, there are a lot of difficulties and delays that occur in practice. And these often lead to, for instance, decisions of choosing a suboptimal molecule that might not make it eventually through clinical trials. So if we can design better, more precisely the molecules, we can also have more drugs that eventually go into market. Okay, so I'm very happy to see that over the last maybe 2 years, there's been quite a bit of progress in this field of protein design using geometric deep learning and generative AI. The list is too long to really detail here, but just wanted to have a few mentions. For instance, RF diffusion is a great method building on top from the Baker lab, which was shown to generate diverse set of proteins that are new, different from everything shown before, and can also bind. And we've also seen the great work by Gainza et al. From the Massif team at the EPFL, creating binders by basically learning fingerprints of surfaces and trying to match them across the PDB. There are other notable works as well. Chroma, very nice work by Ingraham et al., FrameDiff from MIT, and others. Now, these models, even though I really like them, they're not the best for antibody, because usually if you use them, usually won't get antibodies out, and that's for a very good reasons that antibodies are a little bit special, right? If you would have an embedding of all the proteins, antibodies would kind of be at a different cluster because the way they interact with the world and the way they bind with other targets is a bit different. So there's good reason to also think about models that are antibody specific. Things in the field started by looking at ways of designing CDRs. Let me note here, for instance, Refine GNN and Mean as two notable works. And I think recently they've progressed to using denoising diffusion and generative models in order to redesign the loops or the backbone of an antibody. So yes, denoising diffusion dominates as a paradigm for generation. I won't spend too much time here, but denoising diffusion is a way of basically posing a generative model by learning how to go from noise to data, right? And I think everybody here knows about it, so I will go to the next slide. Right. If you look at the literature, there is a kind of a recipe, I think of it, for creating a generative model for proteins and antibodies specifically. So let me sketch it out. There are a few choices that you can make, but this is, I think, we have converged to a pretty consistent solution. The things look like that. So what you do is you represent your protein as a graph where actually the nodes correspond to residues, and you represent the geometry and the structure of each residue. You can do it in a few different ways, but a very common and nice way to do this is through frames, and this came from AlphaFold 2. So here, what you do, a frame consists of one position in space and an orientation. Okay, for instance, we know that using a single frame, we can describe exactly the backbone of a residue. So you present it like that, then you use your SE3 equivariant relational model of choice, and you define a Gaussian a diffusion process that's appropriate for that representation. Usually it's a Gaussian for positions and isotropic Gaussian over the SO3 for rotations. You train it on the most data you can find, and that's it. So this recipe is nice, but I would argue that there are still some limitations, some challenges that we need to face. I think there are two main challenges. The first is that I think there is a trade-off between doing things correctly in terms of the modeling, being faithful with respect to the degrees of freedom and the symmetries, and being efficient. So you need quite a bit of memory to do this, and that often leads to cutting corners in the modeling. Like Common, for instance, approach is to look at a coarse resolution, only look at the backbone of the protein rather than of the entire protein. And this is, for antibodies, this is a little bit problematic because we know that in order to understand their function, you need to know the exact atom placement. So backbone is too coarse. There are others that instead of modeling only the backbone, they first and then filling out the sequence in the side chains, that they do, for instance, a part of the antibody, the CDRs at atomic resolution, they generate it, and then they think about the framework. Which is, is a solution, but unfortunately, we know that there are tight interactions between the framework and the CDRs, because CDRs are loops, and the way that they're supported and they move in space is controlled by how the framework kind of attaches to them. So you cannot design them alone. So this is a problem. The second problem is that we really don't have enough data to train these models properly. So what happens is if you don't take care, these models will overfit. This is just to show you how little data we have. This is these numbers here were taken from eMERGE. If you're very careful on how you look at SubDub. SubDub is the part of the PDB that contains antibodies. Now, if you look at the high quality structures that are unique, there are around 1,600 antibodies and 880 antigens in complex together, which is really not a lot. So how do we go around this? Well, we've been thinking about this for a while, and what we decided to do is to try to exploit the properties of the fold in question in order to ameliorate the current limitations. Right. So actually, the technique can apply to any protein family, but in practice, we only have used it to design antibodies, so I cannot make big claims here. But in theory, the idea should hold. So AbDiffuser, it's the model that comes out, and this is a model that generates the entire antibody sequence structure and framework, CDRs and side chains. And it does use, it's based on diffusion, but it's entirely based on Gaussian diffusion. It doesn't use any of this SO3 business, while still being SC3 equivariant and respecting bond constraints. And actually, what we care about is that it tends to give better quality samples. Now, how this is achieved? Well, the secret sauce is the representation that we're using, which is a fixed length representation based on alignment. And so on top of this, we can have a different architecture that's not relational, actually. And even though it's universal, I think, yes. So it's not relational. It's SC3 universal, and it has the benefit of being significantly more efficient. We can fit 10 times bigger models in a single GPU. A second benefit of using this fixed length representation is that it allows us to better utilize priors. And priors are very important because we don't have data, right? So we need to inject inductive bias into our model. And the third consequence is that it handles sequence length changes very well, because these proteins can be of different size as well. Okay, so let me talk about the representation here. So yes, so all geometric deep learning for proteins is done with basically SC3 equivariant versions of GNNs and transformers. We don't do that. We do something that might seem counterintuitive at first, but bear with me. So this is what we do: is we represent the structure and the sequence as a table. So here's on the right, on the number of rows and columns is always the same. The number of rows is something relates to the number of residues, which is not, but it's not exactly. It's the number of residues after alignment. So there can be gaps as well. And it's always 2 times 149. So 2 chains times 149 characters per chain. And the number of columns is always the same. We have some columns used to represent the sequence, and some columns used to represent the atoms of that particular residue. And we do something very simple. We use MLPs across rows and columns to update. Now, okay, so before you say I'm crazy, let me try to convince you that this is a good idea. So the first observation is that the GNNs and transformers, they are they're built around the premise, especially GNNs, of permutation equivariance, right? We don't care about the ordering. But here with proteins, we do care about the ordering. Ordering is extremely important. Without it, you don't have a protein. So you could actually use a GNN and kind of fix that problem by some positional encoding, for instance. But I would argue that this would be a fix. I would prefer kind of an architecture that would take this into account that would be conceived to take this into account. So the second thing is that a common difficulty that people have, and this is where GNNs are very useful, is that the sizes of things can change, right? So for instance, a graph sometimes can have 10 nodes, sometimes can have 20. So even though the same holds for antibodies, it turns out that there's this easy way to align all of these antibodies so they have the same length. And actually, the way that the alignment looks like, it's much more meaningful. So if you go to antibody engineers and you ask them to tell you if your sequence is good, they will not look at the sequence itself. They will align it first, and then they will tell you, this particular amino acid is weird here. Why did you place it? So one such numbering, there are different ways of alignment, but one such alignment is called AHO numbering, which we utilize here. And it allows the network to see the protein in a different way, because now for every position, so every row in that column corresponds to have a very specific row after alignment. So we know, for instance, that position 100 will correspond to that amino acid that's on the kind of CDR3 that's important to binding. And not only roughly at the CDR3, but at specific location that tends to have that particular entropy and function. So that's very, very useful for the model to latch onto. Okay. Now, of course, you will say, yes, but if you use MLPs across rows, what about equivariance to rotations and translations? Well, it turns out that there is a nice solution for this in the form of frame averaging. So frame averaging is a refinement of the idea of summing over the elements of the group in order to imbue a model with invariance or equivariance. So what happens here is specifically for point clouds, it is almost possible to find a canonical orientation of a point cloud. And the way that one can do this is they can compute the principal component analysis, and they can obtain some vectors, the principal component, and align them with your coordinate axis. Now, if we had a canonical orientation, that would be great, right? Because that fixes our issues. Unfortunately, there is still an ambiguity because we don't know which way the axis of the principal components are facing, right? But it turns out that the number of such ambiguities is not very large. So for the SE3 group, there are only 4 ambiguities. So what we do is we just compute our model 4 times for the 4 different orientations, and we average the output. So, and this is great. It's very simple, very efficient, and has this nice property that if the underlying model that you're using is universal, like we know MLPs are, then also when you wrap them around frame averaging, they remain universal now, but now with respect to the group of interest. So great. We don't have any expressivity problems anymore. I mean, the actual architecture is a little bit more involved. It has, it uses the usual tricks, and it inspired by the MLP mixer. But yeah, I will not spend too much time talking about this. Just to show you some numbers, on the bottom left, what I'm showing is a comparison between AbDiffuser, so this AP mixer architecture, and IGNN and frame average, so GNN wrapped around frame averaging. The number that I'm showing is basically the ratio between Wasserstein distances. Basically, the smaller it is, the better, and ideally you want it to be 1, right? Then you've modeled exactly the distribution. And you see here indeed that this model improves upon the generation quality. On the right-hand side, we also show the number of parameters and memory and generation time required. And you see that it's more than an order of magnitude more efficient, for instance, than EGNets, which is really helpful if you want to model the entire system at atomic resolution, and if you want to use over-parameterized models to, have sufficiently high expressive power. The second thing that I promise is that by using this this representation, one can better utilize priors. So let me explain how we do this. The first prior we use is pertains to the sequence. So I told you that when antibody engineers look at sequences, they look at them after alignment, and they can recognize what's happening. That's because actually, if you would look at on the left of the screen, this is the frequency of amino acids at different positions over the OAS, which is a big data set of sequences with antibodies. And you'll see that this is a pretty sparse matrix. So there is a lot of structure, and many positions, except from the CDRs, are pretty conserved. So what we do is, instead of when we're doing diffusion modeling, using a uniform categorical distribution as the prior distribution for the noise over sequence, what we do is we use this the frequencies that we've estimated from data in order to parameterize this distribution. The second thing pertains to structure. Here, the observation is that usually people use an isotropic Gaussian in the diffusion process, but if you randomly generate atoms, right, they're going to be very even if they are supposed to be close to each other because they're next to each other in the sequence, they will be very far away from each other canonically. Right, so a lot of the work that the generative model needs to do entails actually something that's not very smart about taking these very random points and moving them to something that resembles the protein. And then after this is done, then the model starts to do something more meaningful. So what we said is, actually, there are better distributions that you could use rather than isotropic Gaussian that allows you to take this load away from the model. And what we used here is a Gaussian Markov random field with respect to a learned graph from my friend and colleague Vasilis Kalafolias. Effectively, what this does is it estimates a graph that is sparse and whose covariance matrix can be used to parameterize a Gaussian distribution that describes the positions of the atoms in this case. And this is possible again because we have an alignment. So you can see effectively all the there's a single graph with the same set of nodes across all antibodies, and the only thing that changes are the positions of these nodes in space. Okay, some empirical results. On the left, I'm showing the difference between how reverse diffusion looks like if you have an isotropic Gaussian on the top and Gaussian with a learned covariance matrix on the bottom. And you'll see that so time goes backwards, right? So we start from 1,000, which is completely random, and then we end up at 0, where hopefully you've generated an antibody. And you'll see that at the middle of the reverse process, if you use a unit isotropic covariance matrix, then this still doesn't resemble an antibody, whereas with prior, things look much better. And empirically, we found the use of priors to consistently improve the generation quality both the sequence and the structure prior. And this is across many different ablations and models. Okay, the third part is how do we actually enforce that what we're generating is physical, so it respects the correct constraints with respect to bond distances and angles. Okay, so I told you that the usual way that people do this is by using frames or angles. And if you do this with angles, the benefit is that you can never be wrong, right? So whatever you predict is always going to be a valid confirmation of the backbone. But we have reason to suspect that if you're doing diffusion on Euclidean space, the models may be better optimized than if you're doing it on a manifold, right? So we thought, let's try to find an alternative solution that allows us to use our models in a environment where they're better. We know better how to train and how to use them. So the idea that we had comes from actually constraint optimization, where a way to enforce constraints is to just let in this case, the model do what it wants, and then enforce after the gradient step is applied, you project over the set of feasible solutions using some procedure. And so here, to implement this, what we have is, again, we let the model operate freely. It predicts whatever it wants for the atoms. And then we go in with a differentiable projection layer and we fix any mistake that the model has made. I don't want to go into too many details here, but basically that works pretty well. You can use Gaussian diffusion for atom positions and you get the right geometry at the end. And on the bottom right-hand side, again, I show you the quality of the samples. So the model without projection layer, actually, it was very unstable. So we couldn't train very well. That's why the result is so bad. Whereas, interestingly, with the projection layer, both for the backbone and the side chains, the model started to become more easy to train. So it seems to work better, but we still need to do some work on this. Okay. Let me go through. We have done quite a bit of experiments. Let me go through some of them. So we did things like generate samples from or OES, try to generate mutants from of a known cancer drug for breast cancer that I told you before. That was this is a very nice data set from Mason et al. We also attempted to redesign the CDRs of unseen co-crystal structures. This is a common benchmark in the field. We have a large suite of metrics, both focusing on sequence and structure. And we looked at many different ablations and baselines, things like non-diffusion models, like sequence protein language models, likelihood-based models, different architectures, different priors, and as well as competitive diffusion models and a physics-based baseline. So I think we have a quite a large number of comparisons at hand. The first result I want to give you is that of self-consistency. So here what we do is we check whether when we generate a structure and a sequence, and then if we take that sequence and refold it, in this case using IGFold, how similar is what IGFold predicts with what the generative model gave us. And it turns out that they're very similar. So the RMSD over the whole antibody of AbDiffuser is around 0.49, which is almost the same as the RMSD that the different models in the ensemble of IGFold have with each other. It's 0.42. So it's almost as good as folding, even though it was not trained for that purpose. The second result entails the trying to design new binders for the HER2 target, which is involved in breast cancer. And on the left-hand side, what I'm showing is the the predicted probability of binding for the designs. So what we did here is this set contains positives and negatives. And we took only the positives and we fed them to the generative model, and we asked the generative model to generate more of those. And then we used both positives and negatives to train a classifier. So what we're checking here is whether the generative model can give a consistent answer with as judged by a classifier that has privileged knowledge, knows more about the problem than the generative model. And indeed, we can attain pretty good success rates according to the classifier. Now, when we go to the lab, to the wet lab, the results are perhaps not 94%, but still decent. So if you take the raw samples of the generative model and you don't apply any filtering to them, we found that around 22% bound. So all of them expressed, but 22% bound. And if you use some simple filters on them, basically making sure that the designs were at the best quantile with respect to the metrics that I was showing you before, then the binding improves to 57%. And on the right-hand side, I'm showing the binding affinity. So the higher the number, the better. And you'll see also that here the binding affinity is pretty good, actually, in some cases, even slightly surpassing that of the cancer drug in the market. Trastuzumab. A closer look at the mutants. In this experiment, only the CDRH3 is mutated. So that's why the sequence length is small here. This is the CDRH3. And if you compare the designs with the trastuzumab, you will see that there are around 4 mutations usually. But if you go in the data set and check what are the closest data for which we do have measurements, they're usually 2 distance away, 2 edit distance away. Now, this might be look simple, but actually, if you dig in, you'll find that the closest sequences, they're not to our designs, they're not always binders. So sometimes our designs are equidistant to binders and not binders. So this is, as I was saying, this landscape is pretty rugged. So what we're asked is to generate things that are on the decision boundary, actually, between good and bad molecules. Okay, third experiment, we were redesigning CDRs and in out of distribution fashion. So here we've trained a model to take as an input a complex, delete the CDR with the CDRs deleted, and redesign the CDRs. Right. This is not exactly de novo because you're giving the model the framework and you're giving the orientation of the framework with respect to that target, but still, I think it's a good test. And we use the diff up split, which ensures that the test example that differs from the training. And we use a graph neural network to condition our generative model on the target. The results are pretty encouraging. You'll see, especially on the amino acid recovery, using our methods really help to improve the designs, the quality of the designs, and especially in the CDRH3 region, which, as I said before, is the most tricky one to design. The structure is even improved. So I think it's pretty encouraging. I have a few videos here. On the left, I'm showing how a binder looks like for the HER2. This is for the HER2 interface, the HER2 target. And on the right-hand side, the video is focusing in on the interface. Actually, when I saw this, that's when I started to feel that, we were designing interesting molecules. There's this tendency sometimes to look at the cartoons and to say, okay, the cartoons look very similar to each other, so these must be the same. The problem is not so interesting. But actually, if you with antibodies, you need to look into how the atoms behave in order to understand the complexity of the problem. Okay, so right, I have only, I think, 2 more slides. I think I would like to take a little bit of time to try to convince you that this is a great area to be working on. And there are a lot of open problems if you care about making drugs with generative AI. And the great thing is, so it's a playground for machine learning, I would say. And in this case, actually, if you make progress, this can help cure cancer. I mean, it will not cure it by itself, but it's definitely pushing the ball in the right direction. So it feels good to be working in this field. So from a machine learning point of view, some of the open problems that we've identified pertain, for instance, to learning protein representations, protein language models and structure. Binding affinity is a huge one. Predicting binding affinity, we should make progress on this, and it might be that we need to capture dynamics and entropy to do better. Interpretability is pretty poor, so we could definitely improve on this, and I can tell you that biologists care about it. There's a lack of benchmarks, I think, that allows us to track progress. And if you actually, so in Prescient design, we use a lab in the loop concept where we iteratively, we propose designs and then we get feedback from the lab and then we improve our models and improve our predictions based on this feedback. And that, that creates the whole new set of interesting problems to work on relating to, for instance, how do you choose the designs, active learning, Bayesian optimization, but also how do you handle uncertainty, distribution shifts, and spurious correlations. And there's an interesting paper that recently came out on invariance and distribution shifts. It's called AbDiffuser domain bed that you might want to check out if you're interested in this. Yes, and also how do you create? Because as I said, there are not so many examples that we can train from. How do you bridge machine learning and physics to improve? From my biology perspective, we need to figure out how to select epitopes, like which parts of the target we should go after. The design objectives that we have of expressing and binding, they're too simple. There are actually a lot of other things that matter for creating a lead molecule. Solvent is key. Like, how is the environment of this molecule is going to be look like? How does it influence its function? And how do we better leverage the data from high throughput assays and deal with their uncertainty? I think they're all important questions. Right. My last slide, I think where we are currently is that we can model distributions of proteins and antibodies very precisely. And we are starting to design them for unseen targets. This makes me really happy. It's a great topic to be working on and a great time, I think. What I want to see is that generative AI becomes a part of the antibody discovery campaigns within pharma. And I want drugs to come out using generative AI. And I think that's going to be the true sign of that we made it. Right. There's a list of photos here. This is only a small part of Prescient design. You cannot do this research alone. You need a big team, a lot of talent, a lot of great people. I'm very thankful for teaching me and for helping me. And just let me also mention that we have summer internships available. So if you want to learn more and work on this problem, please drop me an email. Thanks.
Thank you, Andreas. Thank you for the great talk. So we have a few questions. Like the first question, like from Nichols. Do you use any sort of oversampling to increase the size of your data set?
What kind of sampling?
Oversampling.
What do you mean by oversampling?
Like oversampling and undersampling. Is Nico here?
Your connection is a little bit choppy, so I might lose some words. So the things that I know have been done is, for instance, AlphaFold, trains a model and then generates predictions, and they trains a second model based on the predictions of AlphaFold of the first model. which it was always a surprise to me that this works. So self-distillation, but it seems to improve. We have not tested this. Different types of augmentation you can apply, but it's not clear to me that this brings a benefit, especially since we are handling the symmetries pretty well. You could, so the problem is that, okay, so I'm not sure if I'm answering your question exactly, but you can specify if there's something I'm missing. But you can also use physics-based methods to create new examples. But the problem is that these physics-based methods are usually not precise enough to generate very high quality, especially interfaces. So I don't know if you want to be training your model on things that might have error inside them.
Thanks for the answer.
Does that address the question?
Yeah, there are also other few questions, like a question from me actually, like leveraging AP Mixer is actually intuitive, but have you ever tried other model backbones like IPA? If so, can they work?
Yeah, they can work. From our experiments, AP Mixer was so much more scalable, so more efficient, that it allowed us to, it made the training much more convenient. So we didn't have to do distributed training, so I think it was a no-brainer for us. We used we didn't use IPA specifically. We used transformers, compared to transformers, EGNNs, and sequence-based methods, but not IPA specifically.
Okay, so based on your observation, are they like work similarly, or it's just like because AP Mixer is much more scalable than other model backbones?
It's a good question. I think AP Mixer really works on top of alignment, which helps, especially for antibodies, because the fold is conserved. So if you have a single protein family, because we've been talking a lot about antibodies, but you can do this for any protein family that has a conserved fold, like many enzymes have, for instance. You can really exploit that in order to help the model. And if you use IPA, the model needs to learn everything from scratch somehow. You're not exploiting the alignment well. So at the limit of data and compute, probably you can do the same, even though I don't have empirical evidence for this. But in our current regime, I would go for AP Mixer.
Okay, thanks for the answer. So we have also another two questions from Chen Haiyang. The first is that, do you think AP Mixer-like models, for example, GNNs based on frame averaging that are not perfectly symmetry preserving, can also be used for general purpose protein design and generation?
I don't know. It's a good question. I would be a bit more skeptical if your fold changes a lot across different examples. So there, I might think of it twice, but then we haven't tried it. So
Yeah.
I don't have
My
a definite
question
answer.
was more around, if AP Mixer like models are so much more scalable, right? Could it be that just scaling and training for longer can help us learn about rotational symmetries, which obviously seem cumbersome to build into architectures, right?
Well, it is. You are enforcing the rotational symmetry by framing averaging. There are other techniques to do this as well, actually, but so I don't know whether the model would actually learn the rotational symmetry, if that's your question, because it's kind of handled automatically for it. It doesn't have to deal with it somehow. Am I answering your question, or?
Right. Yeah, I guess frame averaging in particular, just looking at different rotations of your input at every layer as well.
Yeah. it's a neat solution, especially if you're doing generation of a single protein family. I think it makes a lot of sense. If you go to different setups, I would think of it twice.
Yeah. My other question was around how important do you think it is to design at the all atom level? I think you touched on this a little bit, right? But to what extent do you think we should trust crystal structures from the PDB, especially for CDR loops and so on? And for instance, the experiments in the lab that you showed with her2, why could we not just have a model which does backbone level design with sequence identities?
Yeah. So you're right that if you have a example experiment like her2, where you are looking at mutations, you can basically encode everything into sequence, and this can work fine. I mean, so I work closely with Kyung-Gyun, who's a big proponent of transformers, and sequence-based approaches. And he always says that, Andreas, everything is in the sequence. That's kind of a perfect representation of your protein. So in fact, you can probably start from sequence and you can ignore everything else. But I think the closer, okay, the closer you get to the structure, and the closer you get to the actual positions of the atoms that will happen during binding, the easier it is for instance, for a classifier to tell you whether what you're designing makes sense or not, right? And so now what does that mean? Okay, first you can screen your designs more reliably if you have a structural hypothesis. But not only that, but because if a classifier cannot really predict whether what you're designing is good or not, how do you expect the generative model to really know that what it's designing is a valid solution or not? So from that perspective, I do think that it makes sense, especially in the de novo case, right, where you have a new target that the sequence patterns are not what are going to be causal for. Not causal, but it's not going to be easy to see from the sequence patterns what the types of bonds will be formed with the target.
Yeah. Yeah, that makes sense.
Very good.
Well, if we don't have more questions from the audience, could you also talk a bit about the challenges of interpretability, or why do you think we need interpretability in this space? You mentioned it in your conclusion, but I'd be keen to hear more about it.
I was never working on interpretability, so but I've noticed that when you're talking to biologists, they want to understand why you say so that something will work, right? That will convince them to trust your predictions, but also you can avoid, like, oftentimes there can be multiple reasons why you're getting an observation, and if you can interpret why, for instance, how your designs bind, they can eliminate some of these possibilities and be convinced that this is a good track.
Yeah. Right. So in a way, like when these generative models and AI systems are working together with real biologists who understand the problem, that's when you need models which can explain themselves in a way.
Right, and this will urge you to, for instance, to do further experiments to discuss, okay, there's this specific mutation that has been identified as key, right? Can we keep it, or can we search for similar mutations so that you can have a further experiment that allows you to capitalize on this intuition? Whereas if you just have a solution and you don't have a clue why this is good, then it's difficult to continue from that.
Yeah. That makes sense. Yeah. Sinyu, did you have more questions as well?
I don't think so. Like, I think all the questions are addressed. Thank you very much, Lucas. Thank you for the great talk. Should we move to the next session?
Yeah, thanks,
Great.
Andreas, for an awesome